Hello, and welcome to this week's Iowa Learning Farms webinar. We are back live and no PowerPoint presentation again, like we did with Dr. Mark Licht last month. I'm Matt Helmers with Iowa State University and Director of the Iowa Nutrient Research Center and faculty affiliate with Iowa Learning Farms. I am really excited, like I was when Mark was here, to go beyond the basics and ask our guest some questions about cover crops and this is an area that I know very little about. I got to see Dr. Alison Robertson yesterday and talk about some of this. And so I think we're going to have a great conversation today about cover crops and plant pathology and why we need to think about this. As always, type your questions in the chat box, but really this week, type your questions in the chat box because this time is yours and we want to get your questions asked and answered. I certainly have some questions, but they're even better if they come from you. So with us today, we have Dr. Alison Robertson. I don't even know what department you're in.
I'm in plant pathology, entomology, and microbiology.
Okay, I'm glad you, so she is extension specialist in this area. So I'm going to start out with Dr. Robertson. Hopefully I can call you Allison after this. Okay, all right. But tell me about your journey with cover crops, kind of where it started, and then we'll get into some of the work that you're doing now.
Okay, so I like to tell this story. So I started it here at Iowa State in 2004, same time as you.
I'm a little
2005, three.
Yeah,
that's right.
Yeah.
I knew we were really close.
Yeah.
And I didn't start working on cover crops maybe until about 2012, 2013, and so I was kind of introduced to cover crops by Tom Casper.
The godfather of cover crops.
Exactly. Yes. Yeah. So he's a USDA ARS scientist for folks who don't know that. And Tom had been working on cover crops for 20 years by then now and was really interested in trying to understand why when we plant corn following a cereal rye cover crop, like sometimes we will get a yield drag, right? It doesn't happen all the time. It happens in some years, not in other years, some fields, not other fields, and it just wasn't consistent. And he wondered whether it could be due to seedling diseases. And I was working on a big USDA project where we were looking at seedling diseases in soybean.
Okay.
And so he said to me, well, what about seedling diseases in corn? And when I looked, when you plant corn following a cereal rye cover crop, like some of the things you see, you're going to see yellowed, stunted seedlings. You might see uneven growth in the field. And those are exactly what you would see with seedling diseases as well, right? And so I thought that Tom had a really good point. And I said to him, sure, I'll work with you. And so, yes, so that's how I fell into cover crops and I love it.
Okay.
So I really enjoy like all the research projects we've been doing and everything. It's just super fun. I've met lots of really cool people. I think the coolest thing about working on cover crops is that I get to work with folks from other disciplines, right? Agronomists, soil scientists, entomologists, weed scientists. And so it's really cool to be able to get out of my little box and do some cool things.
We've never officially worked together.
No, we haven't.
But I think
you're scary to work with.
My God. It was okay. Well, golly, on that note, why would I be scary?
You're an engineer. Engineers are scary.
Drainage and soil moisture seems like it's important as we think about plant pathology. But so let's, I guess, that got me off. That got me off my game in every way, shape, or form. Okay, so as we think about cover crops and plant diseases, I guess what plant diseases are we maybe most concerned with as we think about that?
So on corn, we're most concerned about seedling diseases.
Okay.
And I just want to make a point here, right? That cover crops are not always going to increase diseases, right? There's plenty of examples where cover crops actually decrease diseases. So there's some very nice work with soybeans and white mold, where if you plant a cereal rye cover crop, you actually have reduced white mold in soybeans. So, but in corn, you do have this increased risk of seedling disease. So, yep.
And so what are some of the seedling diseases we might be most concerned about?
So seedling diseases are caused by a number of different pathogens. These will sound familiar to folks, like Pythium, Fusarium, Rhizoctonia. And I guess the serendipitous thing about me getting into cover crops is that my lab does a lot of work on Pythium, on the water molds. And when I started working with Tom, the first thing that we did was we looked at cereal rye, and we wanted to know, does cereal rye, is it a host of these same pathogens? And it was. We could take those pathogens from the rye, put it onto corn, and cause disease.
Okay.
But it wasn't until we did our field research that we found that it's really Pythium that's the problem, and not Fusarium or Rhizoctonia. And so that's the serendipitous part, right? Is because that's what my lab done. And I was like, yes.
Yes. Okay, so I guess one of the things maybe to think about with some of these issues is, and I think you talked about it yesterday, I've heard that it's near and dear to your heart, but describe the disease triangle, because that may help us understand why we see issues some years and maybe why we don't in other years.
Yes, absolutely. So many folks know that I'm obsessed with the disease triangle. And the reason I'm obsessed is because it really explains why we get diseases. So diseases, obviously, you have to have a host and you have to have a pathogen, right? Okay. You have to have, if you think about us, we're the host, and then the flu virus comes along, right? And we get flu. So it's kind of the same with plants. You have your host. So in this case, we're talking about corn. And then you have your pathogen. So we'll be talking about Pythium here, right? But the disease triangle tells us that we have the corn, we have the pathogen, but we have to have the right environment, right? And so for Pythium, a lot of times that's wet conditions. And there are different species of Pythium. Some of them prefer cool conditions, some of them prefer warm conditions, but we have to have wet. That's why Pythium's called a water mold. Okay. The wet is because Pythium will produce these little spores, seeds, that swim in the soil, and they swim towards the roots of the plant. They can smell the plant, and so they swim along and take a chunk out of the root, right? And so, and if we don't have those wet conditions, if we have cool, dry conditions or hot, dry conditions, those spores can't swim anywhere, right? They can't even be produced, and so we won't get disease. And it's like that for any disease we talk about. Last year, southern rust was a big deal, right? And everyone's talking about southern rust. But the spores got here, we had the corn, and then we had the right conditions, and that's why we had the disease problem, right? We see southern rust every year, but because it was so wet last year and warmer than normal, that was just very favorable, and so we got more disease. So so like I go back to the disease triangle with everything.
Okay, all right. Kind of like me with students, they have to take soil physics. You know, they have to understand the disease triangle.
Yes. So I'm like, you're not going anywhere if you don't understand that.
So we got one question here, and I think that kind of starting out thinking about these seedling diseases, and then we'll get talking about cover crops and how we can mitigate any potential risk there. But how can I tell if I have a seedling disease, and is there scouting that I should be doing?
I love that question. Yeah. So, because I think that I was thinking about this morning, right? Like seedling disease on corn following a cover crop is so subtle that I think a lot of people don't realize that they have a problem. Because seedling disease doesn't just mean death of the seedlings and the seedlings are dead. A lot of times the seedling disease that we see is just root rot. So if we think about a corn seedling, the seed germinates, and we have the seedling root system, right? The radicle and the seminal roots. And then we have this pipeline called the mesocotyl that goes to the crown, and then the plant grows from the crown, right? And from the crown, we get nodal roots produced, but those are only produced around about V4 to V6, right? And so up until that plant is at V6, when the nodal roots take over, that seedling root system is very, very important for the health of the plant. And so if we get, a lot of times, what you'll want to do is go out in the field and look at the plants. This is what we do when we go out. And a lot of times, like most of the plants will be around growth stage V2, V3, but then we'll see some plants that are at V1 or V2. Maybe they have a yellowed leaf. Maybe one of their leaves is, the first leaf on the plant is dead. Mm-hmm. You know, that's kind of an indication that you have a problem. You want to dig that plant up and then take the soil off, obviously, so you can see the roots. And what you might notice is like rot on the radicle, or the seminal roots are rotted, or the mesocotyl is rotted. Now, if any of those are rotted, that plant can't take up water and nutrients sufficiently. Mm. And so it's just less vigorous than its surrounding plants. It might survive, but what does that plant turn into if it survives, right?
I think you called it a runt yesterday.
Yeah, well, normally I call it a wimp or a weed.
Yeah.
But yeah, I mean, because it's outcompeted by its neighbors, right? And so it just turns into this pathetic little plant that is there taking some nutrients and some water and producing a tiny little dumpy ear or no ear at all. And if you don't have an ear or you have a dumpy ear, that's lost yield, right? Yeah. And so, so that's what I would do. I would go out, look for those stunted plants, maybe wilted, yellowed leaves, dig them up, and then check the health of the root system. It would be a good idea to check a seedling nearby that looks healthy, because then you can compare the root system, right? And you can say, okay, I see, this has got a much better root system.
Yeah, okay.
So, that's how you would recognize it in the field.
Okay, great. Again, if they, this is a good question that I wondered about. Is there anything you can do to treat the disease if you find it, or is it too late?
Too late.
Oh, okay. All right. Yep.
Sorry, nothing you can do.
Okay. Okay. Yep. So this is why we have to try to minimize the environmental conditions that
Exactly.
Yep. Which I think leads me into thinking about cover crops.
Yes.
And how we manage cover crops. What are some things we can do to minimize that risk?
Yep. So, that's a great lead-in. What our research in our lab is focused on understanding the system, because if we understand the system, then we can come up with management practices. And so there's various ways to manage disease, right? But go back to the disease triangle, right? So you can do something with the host. So you can plant a resistant host. Okay. Now, if you look at a corn seed catalog, they don't really give you resistance to seedling disease. Okay. So we don't really have a good idea.
To do much with the host.
Can't do that with the host, right? Environment, maybe you could do some tile drainage, right? Okay. Reduce wetness in the field and stuff. But then the other thing is the pathogen. So how do we get rid of that side of the triangle? And so the way we can get rid of that side of the triangle is by avoiding the pathogen. Okay. And so we can avoid the pathogen in time, and we can avoid the pathogen in space.
Okay.
Right? And that takes a lot of explaining.
Okay. Well, let's do it. Let's talk about how do we avoid that pathogen first in time?
In time. Okay. So we know that when we terminate the rye with a herbicide, that plant slowly dies. And like I told you at the beginning, the rye has those seedling pathogens in the roots. So what happens as that plant's dying, it releases those pathogens into the soil. Now, those pathogens are always present. They're endemic. So we always have like a certain level in the soil, a baseline. Okay. Yep. But once we terminate that cover crop, we kind of get an increase in the level, and then it'll drop off there. If there's nothing to infect, it'll drop off back to background levels, right? And So when we're avoiding the pathogen in time, we want to avoid that increase in population.
Okay.
And so that's why we recommend terminating the cover crop and then waiting 10 to 14 days before you plant your corn.
Okay.
Because then you're planting into baseline levels of the pathogen. You're not planting into a petri dish of pathogens,
and I think
right?
From the graph yesterday, about 3 days is what
Yeah,
you see.
yeah. About 3, that's not my research.
Okay.
That's someone else's research. But yeah, I mean, we see something similar. 3 to 7 days is when we see the highest populations.
That
and then
petri dish would just
be
full of that.
Yeah.
Okay.
And so that's why we recommend waiting. That's why I'm not very fond of planting green. And I understand that people do it.
Yeah.
And I understand why, right? Because you can't get into the field. And sometimes it's easier to just plant and terminate at the same time. So I totally get.
Yeah.
But that's why we're not big fans of planting green.
Okay. At least for corn.
At least for corn.
Okay.
Yes.
Yeah.
Yes.
All right. Now, that's the time. Now let's tell me about space.
Okay. So the space. So this is research that was done by a master's student of mine, and she started in 2019. So it was 2020, 2021 in the field.
Yep.
And a farmer said to me, "Oh, it's social distancing," and that's exactly what it is.
Yeah.
So when you plant in space, what we did in this experiment is we did not plant cover crop over the corn row.
Okay.
So
where you were going to plant the
corn. Where we were going to plant the corn, right? So we plugged one of the drills, and so we had 3 rows of rye, and then nothing, and then 3 rows of rye, and then we also plugged 3 of the drills. So we just planted rye on 30-inch rows, right?
Okay.
And we found, and so the idea is that the population's going to be highest where the rye is, but then where there's no rye, we're going to have background levels of populations, and we found that really did help.
Okay.
The 30-inch was obviously better than the skip row, just because there's further space between the rye and the corn.
Okay.
But yeah, so that was a pretty fun experiment to do, and at the right time.
At the right time. Appropriate to call it social distancing.
Yeah.
Okay. Now that makes sense. I can envision that. In fact, that's what we've done that kind of on our research site at Gilmore City since 2003, because that's what Tom Casper told us to do at that time,
Yeah.
So, but what about if you're overseed or interseeding and you don't have that ability to plug that? Are you thinking about and looking at strip till to minimize where that rye is growing? And do you think that would have some of the same benefits?
Yes, so that's kind of our next step that we're doing, and we've moved on just from social distancing to now we're comparing social distancing with strip till, and we hypothesize we're going to see exactly the same thing, right? So we don't think there's going to be any difference. I know that there's lots of farmers who already do strip till, and they don't appear to have problems, and so it's just the scientist and us, right, wanting to get the empirical data and verify. Yes.
Okay, we now got another question from online that we'll get to. So, do we need to be concerned with kind of how big the rye is? So, if it's bigger, do we need to wait longer, or that kind
of great question. So, yes, I definitely think so. I don't have really any good data to support that, other than I know, like when she was doing her social distancing experiment, in one year we had cereal rye that was 6 inches tall.
Yep.
In another year we had cereal rye that was 3-4 foot tall.
Yeah.
We saw more disease where we had more biomass.
Okay.
Yep. And there's Stefan Galens from PFI wrote a very nice blog where he looked at the relationship between biomass and yield drag.
Yeah.
And except he converted how much biomass to growing degree days. And there was a really nice relationship there that I want to say it's like no more than 750 growing degree days for rye, and then it should be terminated. So there's definitely this relationship. I'm not sure that anyone's actually,
find it, but something to think about for any farmers out there with us that, especially over the next few weeks, if we have really good growing condition, you know, that maybe we need to be
definitely.
Planting of the corn late.
And I want to say Mark Lick says knee high, and then that rye should be terminated, because otherwise we start getting into problems.
Okay.
So, and I agree with Mark. Yeah.
Somebody asked this, and I'm gonna just I know Mitch answered it in the chat, but we have with us today Dr. Alison Robertson, that's a professor in plant pathology, entomology, and microbiology. Almost got it. Okay. And a field crops extension pathologist. So really great to have her. Okay. So here's one. We're going to move away from corn, but there's a question. You mentioned it. So somebody asked about the cereal rye's ability to help suppress white mold in soybeans. Can you talk a little bit about that?
Yes, that's a really nice, really exciting system to work about. So if you think about white mold, right? White mold, the pathogen produces those sclerotia, those little black rat droppings.
Okay.
And they, excuse me, they survive in the soil. And then what happens is, under specific light conditions, so low light conditions and humidity, they germinate and they produce these little mushrooms, and then those mushrooms expel these spores, which are then blown in the air and they land on the soybean flowers. When the flowers die, they then infect the flowers, right? So with that in mind, what happens is when we have a cereal rye cover crop and we terminate it, we have the correct shading, right? And so those, what happens is those sclerotia germinate and produce those mushrooms earlier and their spores earlier before we have the flowers.
Okay.
Yes. And the same would be true like if you had cereal rye ahead of corn, right? And you've got the corn, but you maybe had a white mold problem the year before, those sclerotia would still germinate.
Yes.
And the spores would be expelled, but there's no soybean flowers, right? There's only corn.
Yeah.
And so, so it would just help reduce the inoculum in the field.
Yeah.
So, and it's, I think it's just a really cool system. Yeah, it's so awesome how biology works.
Kind of get at that, you know, we've been talking about kind of the problems here, right? Yeah. But this is, you know, why would we plant cover crops? It seems like, well, we have all these problems, but here's an example where it might provide some benefits. We definitely know that cover crops help us with soil erosion and nitrate leaching. So there are a lot of benefits, but you know, kind of how we manage this for corn. I think you've said corn is not it's not the hardiest of plants. I think you might have called it princess. So, you know, I don't know. My goal today was to get you to say that corn is a princess.
Corn is a princess. Right. But I was going to say something else, Matt. What was I going to say along those lines? Corn is a princess. Oh, and I always say the benefits of cover crops far outweigh the negatives.
Okay.
So I firmly believe that, right?
Okay.
Yeah.
Great.
And I think that we can manage those negatives, right? Yep.
Yeah. And so, that timing, if we think about that timing and space, social distancing in both. Yeah.
Yep.
Is there any research being done on disease transfer between cash crops and other cover crop species? I guess you mainly worked with rye.
Mainly worked with rye. We did a little bit of work in the greenhouse, where we looked at barley and wheat and other small grains, and triticale, we saw the same effect as the rye. We've done some work with camelina.
Okay.
And there is other work, like I know Leonor Leandro and Greg Tilker have both done work looking at other cover crops and how they might impact soybean cyst nematode and seedling diseases of Pythium. Yep. So there is work on that. At the moment, I've just been focusing on cereal rye because that's where the problem is, right?
And that would be the one that primarily that's widely used and over
80%, I think, is what we learned yesterday from Ethan, right? 79% of Iowa growers use cereal rye. Yeah.
And if we have something that doesn't overwinter, we would not expect that to happen.
Oh, no. So with oats, if you plant oats, you're not going to have, it's called the green bridge effect, right? Where we have the pathogens from the rye jumping onto the corn, with that time. So that wouldn't happen with oats because oats is, dead. Yeah.
So I asked you a question before we started today, whether because you talked about termination and herbicide termination. Does it matter what herbicide we use? Because, would that impact, when it's brown? Because I think you said you want to plant brown.
Brown, yes.
And so,
Yep.
Are you doing any work looking at different herbicides and how that might impact this?
Yes, we are, because there are reports in the literature that glyphosate will increase soil-borne pathogens. Okay. But I also know from Megan Anderson's work, right, Megan, field agronomist here with Iowa State, that glyphosate is by far the most efficient way to terminate cereal rye with a herbicide. And remember, you can terminate cereal rye by roller crimping, but you can't do that with corn, right?
You can't,
because then you're planting your corn.
Mark talked about that on roller crimping. You got to wait so long.
Yes, exactly. So we can't do that with corn, so we have to use a herbicide. So right now what we're doing is we're comparing glyphosate and paraquat, and we want to have a look at the effect of those two herbicides on disease and then on the Pythium populations as well. And I can't quite remember Esteban. He's doing his PhD on this. I can't quite remember his results, but I want to say that we have not seen a difference in disease, but we might have seen more Pythium following glyphosate. But that was only 1 year of data. So that was 2024. I haven't seen the 2025, and we'll repeat it in 2026. But we are looking at that. The problem with paraquat, though, is that you don't get as good as termination. Okay. So you might get, second growth, comeback growth. I don't know what regrowth. Regrowth. Yeah. And but we're looking at it.
Okay. Excuse me. Okay. Are there any, you talked about some of the things you're looking at. Is there anything else that you're starting to look at in research that you're super intrigued by?
So another thing that we're doing is a lot of people have pointed a finger at allelopathy for causing this yield drag. Yeah. And so we're working with Marshall McDaniel on this and trying to figure this out. And allelopathy is really hard to figure out. But so we did a little bit of work in the lab where we took some just synthetic allelochemicals, put them into a petri dish with the pathogen and the corn seed, and we got more severe root rot. Okay. And so, allelochemicals usually, the weed scientists really like them because they prevent germination of weed seed, right? Yeah. Which is very small seed. Yep. And now we've got this honking great corn seed, right? But in the petri dish, the corn seed just didn't grow as well. And so our hypothesis is that rye is producing allelochemicals and they're just weakening the corn seedling, and so it's more susceptible to seedling disease. Okay. So what our field trial is that we have 4 different varieties of cereal rye, which, according to the literature, produce different amount of allelochemicals. And so we have those out in the field, and we're looking at, differences in disease, and we're collecting soil samples to look at the allelochemicals. And I can tell you from 2024 that we saw differences in disease among the varieties, the cereal rye varieties, right? But we did not see differences in allelochemicals. Okay. So that was 2024. I haven't seen the 2025 data yet. So, yeah.
So the allelochemicals, I wouldn't say this right.
I don't think I'm even saying it right, but I talk funny anyway. So
do you think that I guess the social distancing in space would help that too?
Absolutely. Okay. Yes. Yeah. So I think it's like a combination.
That these don't move that far?
No, they don't
soil?
move very
Okay.
far, and they don't last very long in the
Okay.
soil either.
Okay.
So they break down really quickly, like within about 10 days.
Okay.
So it
This
lines
may
up
help.
perfectly with
Okay.
the populations, right?
Okay.
And so it's like, are we looking at chemicals, or are we looking at Pythium, or are we looking at an interaction?
Okay.
Yeah.
All right. All right.
Yep.
Okay. So we had some questions about a question about best options and timing for cover crop into soybean for weed control in late season. And that one, we are going to say, tune in when we get Dr. Wesley Everman. I think he's on the hot seat next month. And
Okay.
so he'll talk about that, and it'll be a perfect time because, it's kind of right before we might start terminating before soybeans. So please tune in there. And I think it's not really in Allison's wheelhouse on potential nitrogen gain rotating to corn the next spring, but I think that'll be something that a future topic for this type of format is getting somebody that can talk about the nitrogen dynamics.
Yep.
This is great. Last time we had somebody bring something up with manure, and so we're going to get Dan in here, Dan Anderson, to do this. And this question about nitrogen makes me think of getting Dr. Richard Roth in here. So please tune in and keep your eye out for more of these in this format. So does the time that maybe the field has had cover crops, do you think that impacts any of these seedling diseases?
That's another great question. So no, I don't think so. And the reason why I don't think so is because when we started working with Tom Casper, we were working on his plots, right, that he'd worked on for 20 years. Soybean, corn rotation with cover crops, no-till. And so, I think that I don't think it has anything to do with the length of time that you've been in cover crops.
Have you looked at kind of, I guess, any soil moisture impacts? If we have a cover crop, we might reduce soil moisture, especially when that cover crop is growing. We could then increase it, maybe if there's a mulching effect. Have you looked at that at all? Or
so I remember in our early work, we definitely had temperature probes in, but I'm not sure that we had moisture probes. But yeah, man, I mean, those are all really good questions.
Yeah,
right?
I think
because
I'm wanting to, see how we can interact with drainage impact.
That's awesome. Yes.
I'm thinking about, soil moisture and drainage and this. There was a question in here about, that paraquat is not widely used. And so, kind of maybe why are we doing research on that when maybe it's not a widely used chemical, or there's maybe, I think there may be more issues with it.
That's a great question. And I guess we're doing research on it because sometimes, I agree, it is ivory tower research, right? I mean, paraquats are horrible chemical anyway to be using, right, for a start. And so there's no ways that I would recommend farmers use paraquat. It's just, we're just using it to try and understand the system.
And
right?
understand how different herbicides or modes of action maybe are impacting.
Exactly.
Disease,
so it's that I would definitely not develop a recommendation on that. Yeah.
All right,
that's ivory tower stuff.
Sometimes we have to do a little.
Yes, exactly.
Right?
Yeah. So, unfortunately,
I think we still have time for questions if people have them. So I think you've talked a lot about Pythium.
Yes.
And Fusarium, and what's the third?
Rhizoctonia.
Right, and those maybe are less of an issue as we think about this.
Yeah. So what we found was that rye does host Fusarium, and that Fusarium does cause disease on corn. But when we did our field trials, what we found was that after where we had seedling disease, so we would take those seedlings to the lab, right, and then we would look what pathogens were present on the seedlings, and it's usually a mixture of Fusarium and Pythium.
Okay.
But where we had the cereal rye, we had more Pythium compared to no cereal rye.
Okay.
Where we had Fusarium, there was no difference between no cereal rye and cereal rye. So that's a treatment effect, right? That indicates that the cereal rye is increasing the pythium, but it's not affecting the fusarium.
Okay.
Yeah, yeah.
So I guess if we circle back and think about some of the things to summarize here is 10 to 14 days after termination of that rye, we should be able to mitigate that risk with seedling diseases. And kind of also thinking about that space of trying to keep where we're planting that corn a bit away from where that rye has been growing.
And that could be done with precision planting or with strip till.
Okay.
And the other thing that I should bring up that we haven't talked about is that we actually did combine timing and spacing. So we just finished that work. I did that with Peter O'Brien, who took over from Tom Casper, and where the publication is in the works. But we were able to show that if you plant green and spatially distance at the same time, so you can use spatial distancing to reduce the negative effect of planting green.
Okay,
does that make sense?
Yeah. So that if you're using the spatial distancing, you don't have to worry about the temporal distancing.
Yes, exactly.
Okay. So that could be a really good kind of option for somebody that wants to let the cover crop go a little bit more. Also, as we think about people pushing planting of corn earlier, it seems that we run into some problems in years of having good herbicide. This would be something for Dr. Everman, but that there's a little more challenge sometimes with terminating the rye with glyphosate with temperatures. So if you can spatial distance, let that rye grow, get a little warmer conditions, and then terminate it.
Exactly.
Another real benefit of strip till as we think about it in a lot of our So maybe we'll go to anything else that you want to share? We have time for some questions here, but anything else?
I don't think so. I mean, the work that we did with Peter, where we combined the two, the planting green and the spatially distanced. Another thing that we did look at there was things like erosion and infiltration, because when you're precision planting or strip tilling, you're leaving less cover crop on the landscape, right? And the reason why we're planting the cover crop is to take up that nitrate for the soil health benefits, to reduce the erosion and everything. And so we did some small plot trials with him, and the data was very variable, but we were able to show that there wasn't really an impact on erosion or infiltration, anything like that. So even though we're planting less cover crop on the landscape, we're still getting the benefits of the cover crop.
Mainly the benefits that we would expect and want to get for those thinking about what are the benefits of cover crop. So I think we'll have time if folks have questions, but we'll go to kind of the last some slides here, logistical items. So I want to really thank our key partners that make the Iowa Learning Farms Conservation Webinar Series possible: the Iowa Department of Agriculture and Land Stewardship, the Iowa Department of Natural Resources, the Iowa Nutrient Research Center, the Leopold Center for Sustainable Agriculture. And if you watch this webinar live and need to earn a CCA CEU, please email Elena Whitaker. Not please. If you want to earn it, email Elena Whitaker. Email Elena Whitaker, and her email is alenaw@iastate.edu. Email her today by 5 o'clock and include, that should say Wednesday, March 25th. So we had the same So by Wednesday, March 25th, by 5, your name, the name you entered to watch the webinar, and your CCA number. Again, alenaw@iastate.edu. Let's go to the next slide. Okay, if you watch this webinar live and are willing to fill out a voluntary demographic survey, please, we greatly appreciate it, okay? Only one survey response is needed for all 2026 webinars. You can scan that QR code or go to that link, iowalearningfarms.org/survey. Again, only one response is needed for 2026 webinars. Okay, let's go to the next one. Okay, we're going to go next week. And for the webinar, we're back to our
PowerPoints.
That's right. So Honeybees, Friends for Conservation. Oh my gosh. Friends for Agriculture, Foes for Conservation. So Dr. Amy Toth, is she in your department? No, she's in
She's in
EOB. Yeah, okay,
yeah,
sorry.
yeah.
Yep. So please tune in to hear about honeybees. And okay, if you watch this webinar live and you want to go back and watch it, it's archived. If you know somebody that might benefit from watching it, please send it to them. They'll be on the Iowa Learning Farms website or on the YouTube Iowa Learning Farms webinars page. And I think, do we have a plug? Oh, tomorrow at 1, there is a virtual field day. So if you're on and want to hear from Dr. Charlie Lowen at Iowa State University, talk about wetlands and looking at some of the benefits and any unintended consequences of wetlands, please tune in tomorrow. I think with that, somebody said, tell Alison she did a great job.
Thank you.
Yeah, that's right. As long as our guest does a good job, the host doesn't have to do much at all other than just prompt her with questions. So Alison, thanks so much for the work that you're doing. If I put on another hat as INRC director, thank you for all the great work that you're doing in this space. I think for farmers in Iowa, this work is super important and is helping mitigating some of the risk of cover crops. Yeah. So
thanks for having me on.
Yeah,
it was
thanks
fun.
so much. And again, we'll be having Dr. Wesley Everman in this format. So get those questions ready about weed management and cover crops coming up. I don't have the date. I can't remember what I'm doing tomorrow. What's April 15th? April 15th. Okay, tune in April 15th, day after tax day, right? So, yeah. Okay, thanks very much.